first session.
Good afternoon, everyone. Myself, Dr. Aritra Kumar Ray, is Senior Resident, Department of General Medicine, Aritra Medical College and Hospital. And my topic today is haptens. Now, to begin with, let me explain some introductory terms. Now, what is immunogen? Immunogen is a stimulus that produces a humoral or cell-mediated immune response. Antigen is a molecule that can bind to an antibody, B-cell receptor, or T-cell receptor. Now, the point to be noted here is that all immunogens are antigens, but not all antigens are immunogens. Now, what is epitope? Epitope is a small area on the antigen, usually consisting of 4 or 5 amino acid or monosaccharide residues that interacts with a single antibody molecule. Now, what is paratope? The combining area on the antibody molecule corresponding to the epitope is called the paratope. Now, coming to our topic. Now, what are haptens? Haptens are low molecular weight molecules, the small organic molecules that are antigenic but not immunogenic, which means they can bind to immune cells but fail to induce humoral or cell-mediated immune response. Hence, no antibodies are raised against haptens. Now, the term hapten was given by the immunologist Carl Landsteiner, who studied them in the early 20th century. It came from a Greek word hapten, which means to fasten. There are two types of haptens, complex and simple. Complex haptens are relatively large molecules which combine with specific antibodies forming visible precipitate. Example, capsular polysaccharide of pneumococci, cardiolipin. Simple haptens are low molecular weight simple chemical substances, and when they combine with specific antibody, no precipitate is produced. Now, as we can see from this slide, a hapten cannot function as an immunogenic epitope by itself, but when multiple molecules of a single hapten are coupled to a carrier protein, the hapten becomes accessible to the immune system and can function as an immunogen. So why haptens are not immunogenic? Haptens are not immunogenic because they cannot activate helper T cells. Failure to activate helper T cells is due to their inability to bind to MHC proteins. Haptens are univalent, hence cannot activate B cells by themselves. As we know, haptens can induce immune response when covalently bound to a large protein called a carrier protein, forming immunogenic hapten carrier conjugate. Carrier protein molecules can be both antigenic as well as immunogenic. As we can see from the slide, a hapten by itself cannot lead to B cell stimulation. But when a peptide of the carrier protein is presented in association with class 2 MHC protein to the helper T cells, the activated helper T cells then produce interleukins, which stimulates the B cells. Now, as we can see from this slide, haptens by itself do not activate B cells, but when haptens bind to bovine serum antigen, it leads to activation of B cells. Now, in this slide, we can see animals immunized with such a hapten conjugate produce antibodies which are specific for the hapten determinant, unaltered epitopes on the carrier protein, and new epitopes formed by the combined parts of both the haptens and carrier. Now, this is the slide demonstrates the pioneering work of Carl Landsteiner, where we can, I will explain this slide in the next slide. Now, as we can see from this slide, when a rabbit is injected with hapten, there is formation of no antibodies. But when a rabbit is injected with a protein carrier, anti-bovine serum antibody is produced. And when a rabbit is immunized with hapten carrier conjugate, it leads to the formation of anti-DNP, anti-BSA, and anti-DNP-BSA antibody. Now, Landsteiner immunized rabbits with a hapten carrier conjugate and then tested the reactivity of the rabbit's immune sera with that hapten and with closely related haptens coupled to a different carrier protein. He was thus able to measure specifically the reaction of the anti-hapten antibodies in the immune serum and not that of antibodies to the original carrier epitopes. Landsteiner tested whether an anti-hapten antibody could bind to other haptens having a slightly different chemical structure. Such reactions are called a cross reaction. Now, coming to some of the examples of haptens, some of the examples, dinitrophenol, orthoaminobenzoic acid, M-aminobenzoic acid, para-aminobenzoic acid can act as haptens. Many biologically important substances which can function as haptens are drugs such as penicillin, peptide hormones, and occasionally steroid hormones. Now, coming to the fact that what are the application of haptens in real life? The formation of a drug protein conjugates in the body can produce drug allergies that can be life-threatening. Immunologists favor it as it can be used as a diagnostic tool and enzyme multiplies immunoassay technique. Now, coming to the application of haptens in pregnancy test, we have to collect the urine from a mother and anti-HCG antibody is added and incubated and incubated. Then we have to add HCG carrier conjugate, and we have to observe for any visible clumping. Now, what are the possible reactions? When a mother is not pregnant, the anti-HCG antibody binds to the HCG carrier conjugate and visible clumping is observed. But when the urine of a mother contains HCG HCG, the anti-HCG antibody binds to the HCG in urine and no visible clumping is observed. Previous trials have also demonstrated the role of hapten carrier conjugate in the treatment of drug abuse and also in the treatment of cancer. But many trials are needed in order to evaluate new horizon, and new aspects are yet to be found out. Thank you.
Thank you.